All right, everyone, we're going to get started. Today we have Dr. Pete Rand talking about the migration study that he did on Sockeye salmon. Pete works at the Science Center, as many of you may know. Okay, Pete, here.
All right, great. So just want to start by acknowledging Kristen Gorman, who many of you may know. She was here at the Science Center for a number of years and then went on. She's now a faculty member at the University of Alaska Fairbanks. And she was actually kind of got this whole project started on the Copper River through some conversations she had with gill netters in town. And I think many of you know there's been this kind of this long-term decline in size of adult sockeye coming back to the copper, and there was a lot of concern about having low catchability. These fish are getting small enough that they're passing through the mesh in the nets, and that could be affecting their ability to catch fish on the flats. And so Kristen's done a lot of work with seabirds in the past and has thought a lot about physiology and stress in organisms, and I've done a lot of work with fish and salmon. And so we talked over the years and did some pilot work in 2016, and then this sort of led to a larger project that I'm going to present to you today. And so it's just important to recognize and acknowledge that we've got some of these initial ideas from the fleet, from the commercial fishing fleet, and it's really a nice place for scientists like me to work because I get to interact with a lot of stakeholders, a lot of fishermen in the area. And this project has also given me a whole different perspective on the copper by traveling up in the headwaters and getting to know people, native people, and other people that are studying the fish and the fisheries. So it's been a really, really interesting project. And so I'm going to kind of move through this. If you have any questions, hopefully we'll have time at the end for questions, but stop me if something is unclear. I just wanted to acknowledge where we work. We work on native land, so this is, of course, Eyak, Lutic, and Atna tribes, particularly in the upper part of the watersheds. We recognize, appreciate, and honor Indigenous peoples. We're committed to building relationships with Indigenous organizations, and we've been working very closely with Atna Intertribal Resource Commission out of Glen Allen. The idea is to create relevant and actionable information in support of their sovereignty and self-determination. So this is a picture taken in the one of the, well, I'll talk about this a little bit later, Mentasta. This is right outside of Mentasta village on Mentasta Lake. This is one of the sockeye populations that travel the longest distance to get to the spawning grounds. Okay, and everyone is interested in where we're getting funding, so we just wanted to acknowledge a lot of it is federal support through the North Pacific Research Board, through NOAA, the Alaska Sustainable Salmon Fund, that supports a lot of projects, particularly for fish and game in the state. Alaska INRI, which is based at the University of Alaska Fairbanks, biomedical research applications to a lot of different human disease issues, but also applies to work with wild animals and fish. And so they've helped support some of our work. And then, yeah, it's important to acknowledge Copper River Marketing Association, and most recently the National Park Service. And I'll describe a little bit more about how they're supporting our work. So a lot of different sources going back to 2016 when we started this work. Here's just a rough outline of what I'm going to be covering. Basically, there's changes happening in the ocean and in the river, saltwater and freshwater, and this is affecting salmon at all different stages in their life history. I'm going to be focusing on the adult migration stage. in the river. So I'll introduce the study. There's basically two different approaches we're taking. We're looking at the composition of the body in terms of energy. We're also looking at health metrics and a variety of other things. I'll go into detail on what we're measuring. And then we're capturing the fish in the lower river and inserting radio tags and tracking them upriver. So this has been done a lot of different river systems. It's been done in the copper by Native Village of Eak and LGL in the past, and they're continuing to do work on Chinook salmon, but we've taken a slightly different approach in our study, and I'll describe that. And then future directions, and then hopefully we'll have some time for questions at the end. Yeah, I think a lot of people are maybe tired of hearing about the blob, but this is these unusual marine heat waves that we've been experiencing. This is a map of the globe, and the color here indicates warmer than average temperatures. So if you see the orange and reds in this picture, it's about 5 degrees centigrade above average. And this is what was referred to in media as the blob. And this developed early in like 2013 and became most intense in 2015 and 2016, and extended from the Gulf of Alaska all the way along the west coast of North America. And that's sea surface temperature, but it's important to also recognize that it went deep too, so about 300 feet deep into the ocean. The water temperature was much warmer than average. The nutrients were lower. It had a lot of impacts on the food web, seabirds, mammals, fish. And it clearly had an effect on Pacific salmon as well. So these are expected through global warming to become much more frequent in the future. So this is basically a real concern for fish and fisheries moving forward. We're seeing this same warming signal in the Copper River. This is Alaska Department of Fish and Game data. They've been putting water temperature loggers on the north and south bank at Miles Lake. And I'm showing the trends in temperature in, this is the north bank and this is the south bank. And the trend lines here indicate that the temperature is warming. It's still well below what temperature would be stressful for salmon. But it's about 0.8 degrees Celsius increase over this period. The record starts in about 2006 and goes to 2020 here. So slightly higher rate of increase in the north bank compared to the south bank. South bank is about a ½ degree increase over that time period. And this is the north bank basically is reflecting this, what's happening, this warming trend that's happening in the upper part of the river, whereas the south bank reflects a lot of the water coming out of from glacial melt from Miles.
What constitutes the vertical axis or axis on the graphs? I mean, that's obviously water temperature, but it must be a time series or something.
Yeah,
Come up with all those different.
Yeah, so these are just temperature records over the month of July. That's the total period that the sonar is operating in the river.
Oh, okay.
Yeah.
So June and July.
June and July.
Yeah.
4 degrees a day, are you seeing?
That's over the course, so this is a range over the course of the whole June, July period.
May, yeah. Okay.
At $1 Million Bridge at Miles, they also, USGS measures discharge, as many of you know. So this is the mean discharge in the month of July, which is when many of the fish are passing through. And you see a trend of increasing discharge. So when you think about climate change and salmon, a lot of people think it's leading to warm temperatures and drought. And lower flows, but in the copper you see the reverse. So because of increased glacial melting, we're seeing much higher flows. And many of you probably remember in 2019, record high flows and flooding in the lower copper. And so it's much higher than what you would expect to see if you extended this trend line. So I'm going to be talking quite a bit about this year. So this is the first year where we started the telemetry work, and our results were very dramatically different that year compared to 2020 and 2021. This is just a mass balance. This is the rate at which glaciers are disappearing through south central and southeast Alaska and in the interior. And you see many of these areas in orange and red are glaciers that have negative mass balance or are losing, melting, and getting smaller. And so these warm temperatures are leading to higher flows in rivers like the Copper. So 2019 was this major heat wave that occurred, and it caused a lot of wildfires in the interior of Alaska and very, very warm temperatures. And we summarized mortality events. So these are salmon that didn't spawn, pre-spawn mortality events, mostly visual observations from people in a variety of different river systems, over 100 cases of all the species found dead, mostly due to warm temperatures, but also low flow conditions leading to hypoxia. And you'll see all of the species were observed across the state, going from the Arctic Yukon-Kuskokwim area to Bristol Bay, Inlet, Prince William Sound, and only one observation we were able to document in southeast Alaska. But this basically indicates kind of changing conditions where salmon are getting increasingly subjected to these conditions of warmer water and low flows. But of course, in the Copper, we saw something very different. What I'm going to be talking about. This is the declining size. This is commercially caught fish on the flats from 1960s to close to present. And this is a size at a given age, and you see a declining trend. It's about a 7% decline in length over this time period. And this is kind of what initiated a lot of this early discussion about what this might mean for the fishery and what it might mean for fish coming back smaller. There's a lot of different theories about why these fish are getting smaller. Certainly these heat waves have had an effect, probably a negative effect on their size, but also it's competition in the ocean. So sockeye are known to compete for prey resources. They feed on a lot of the same prey items that sockeye salmon feed on. And so I'm working with Greg Ruggiero, who's a scientist out of Seattle. He's put together a lot of information on growth rates of sockeye based on scales. So they're able to estimate the distance between circuli laid down on scales. The wider that distance, the more the fish grew in a given year. And so we had about 30 years of data from seven different populations from Bristol Bay all the way down to southeast Alaska. These are adult scales that were taken from fish that returned, and we were able to relate the rate of growth of the sockeye to the amount of competitors in the form of pink salmon in the ocean. And you see a negative trend in all of these cases. So in years of high pink salmon abundance, you see lower growth rate. And that explains about half of that trend line that I just showed you. So about half of that decline is explained by the numbers of pink salmon in the ocean. So that's an important pattern that we should be aware of as well. So not only the temperatures and ocean conditions are changing, but the amount of competitors in the ocean are also changing.
What, over on the peninsula, over in Russia there, is that, is that the same? Did you get the same results over there?
Oh, yeah, I included Russia in here because you'll notice that we're relating this to pink salmon originating from both East Kamchatka and Alaska. And so a lot of the pink salmon that sockeye salmon are competing with originate from Russia. And so they're all, they basically all share the same ocean habitat, both in the Bering Sea and the Gulf of Alaska.
So are those pinks coming from Kamchatka, but are there pinks that are raised on Sakhalin Island too, or is that, it's a different species?
No, they're also raised. These are all wild fish from Kamchatka, but there's both wild and hatchery fish in Sakhalin Island as well. But the relationship is strongest when you just look at East Kamchatka pink salmon and Alaska salmon. They're all, basically, sharing the same prey resources in the ocean. So here's our basic research question. What factors may be related to mortality risk for river migrating sockeye while en route to the spawning grounds? And trying to understand what are the implications of fish coming back smaller. And so we're looking at size of the fish. We have a couple different ways of measuring how much fat or energy is in the body of the fish. We're looking at the different spawning stocks or different spawning populations. Each have a unique run timing. So we're looking at early, mid, and late run fish. We're looking at disease and pathogen presence in the gills of the fish, in the gill tissue. And we're looking at gene expression, looking at genes that are related to stress, and trying to understand if that's a good predictor of their ability to migrate successfully. I'll touch a little bit on this idea of comparing the different populations, looking at ones that travel long distance versus ones that travel a shorter distance, and looking at this trade-off in terms of energy and investment in eggs and reproduction. And then basically these are the two components that I already talked about, body composition of adults and radiotelemetry. So I'll give you a little more detail on how we did that. This probably is not needed in this crowd, but I'm just important to recognize the life cycle of these fish. You start in eggs and gravel in 1 year, and then they overwinter as alevin, they emerge as fry, and then they do all different things based on the species and their location. Most sockeye migrate into the lakes, and they rear in the lakes for 1 year or 2 years. Some of them are river sockeye and don't spend any time in lakes. They migrate to the ocean, and then they come back as adults and close the life cycle. There's Sean. Sean Donato helped me 1 year in a lot of different aspects of the project. He's basically doing some of the sampling. This is at Mancasta Lake. I mentioned this previously. So the idea was to collect fish in the lower river when they first enter the river, and in the upper part of the river close to the spawning grounds. So we collected nearly 500 fish and about 150 per year. It's spread out. I think we had 6 or 7 different spawning sites that we visited. And we tried to limit the numbers of samples to just 10 fish, so 5 males and 5 females. So we reduced the impact on these populations, some of which are relatively small. And these are the things that we measured: length, weight, determined sex of the fish. We measured energy content with this fat meter that I'll describe in the next slide. And then we also took these gill samples, gill tissue samples, to look at disease and gene expression, which I'll explain in a little more detail. This is this fat meter. This was actually developed in aquaculture as a quick, easy way of measuring fat content based on microwave sensor. It's actually measuring moisture content, but that's inversely related to the amount of fat in the fish, and you can use it on live fish or dead fish. This is Eric Wong, another one of our technicians that assisted in the project. And he's basically holding the sensor, so you put it right up against the fish and you press a button and it gives you a measure of fat content. And it can range from less than 1% to as high as like 15 or 16%. So we're able to do that on fish at different points in the migration and for different populations.
Where's that slide taken?
That's in
Tanada Lake.
Tanada Lake, yeah. This is just some images of, some of the fish that we sampled. This is Sean and Michio, a Japanese colleague of mine that helped one season. These are the gill clips, so they're placed in this special preservative. This is a large sample taken on a fish that we sacrificed, but for live fish, which I'll describe, it just requires one or two of these small filaments. It's a very small sample taken, so it doesn't harm the fish. And so that's the body composition work. And then just the next couple of slides show some of the patterns that we found. And you probably know the early fish that come back to the river travel the farthest. And so it's not surprising that fish that arrive in May have the highest fat content. And then fish that arrive late July and August have lower fat content. This is megajoules per kilogram. So this was measured. I mentioned there's two ways of measuring energy content. We measure one in the lab based on calorimetry. And then we relate that to those fat meter measurements that we used that we made in the field. So this is actually, these are measurements made in the laboratory on fat content. And I should mention that the whole fish is basically ground up and we take samples, dry them, and then estimate the amount of energy in the sample. So in terms of energy provisioning, the early fish have much higher fat than the late run fish. And these are some of the samples that we collect in the upper river. I'm going to show you some of the patterns, some of the estimates of energy content from these fish in the upper part of the watershed in the next slide. But this is Long Lake in the Chittenden drainage. We have two streams in Clutina Lake, Malo in St. Anne's, Golcana River, and Fish Creek in the upper Golcana and Lantasca, and there's another lake site. Okay, so I just showed you the lower river data. This is the upper river data. So these are the same units, megajoules per kilogram. Each column here is a year. This is 2019, 2020, 2021. These are the Bear Canyon. This is the lower river. And then all of these are the upper river sites. And the bottom line is we measure about half of the amount of fat in these fish that are just arriving on the spawning ground. So they use about 50% of their body energy to get up to the spawning grounds. There's one real outlier here, Long Lake, which they come in very late, and they come in with much more fat than all the others, about 50% more fat. And there's a very interesting difference in their life history compared to all of these other stocks. And I'll talk about that near the end. But that's the Long Lake, that's on the way to McCarthy on the Chittenden River tributary. These are some results from the gene expression work. So this has been really long delayed because of COVID. The laboratory closed down. It had just recently reopened, so we had to wait a long period of time for these results. This is Christy Miller's lab. This is the Department of Fisheries and Oceans in Canada. And they've been looking at these issues about stress in salmon, particularly in the Fraser River in British Columbia. This is the first kind of data that's been worked up for Alaskan salmon. And this is a threshold gene expression. So this is each point here is a fish, a result of a fish. And this is a panel of genes related to viruses. So this is fish responding to disease. And this is a thermal panel. So these are expressed when the fish becomes stressed due to warmer temperatures. And this is year. So we have 2019, 2020, and 2021, the 3 years of the study. And so we got results that make sense to us in terms of 2019. These fish were clearly stressed, and it looks like they were responding to disease. So all of these points, many of these points here were above this threshold, indicating that the fish was diseased and stressed because of a virus. And then this is the response to temperature. And as you would expect in the lower river, these were all fish sampled in the lower river, that very few of these fish showed a stress response to temperature. And we found somewhat counterintuitive results here with 2020 and 2021 having some fish responding to or being stressed due to higher temperatures. So we're still trying to understand how to interpret these results. And it may be just a differences in the genes that they're examining in the Fraser River versus genes that might be important for describing stress in the upper river. So moving on to the other method that we employed in the study, which is radio tagging. So over these same 3 years, we worked with Native Village of Eak at the fish meal site in the lower river, collected fish, tagged them, made a lot of these measurements that I just described on the live fish, released them, and we were able to track them up river with the help of Fish and Game. This is the Canyon Creek fish wheel and some of the Native Village of Eak staff that assisted us on our project. And so 1 year we worked out of Canyon Creek, the other 2 years we worked out of Bear Canyon down river. And this is our basic approach. We collected the fish, and then we made a lot of different measurements on the fish size. We measured fat content with our fat meter, and we handled the fish minimally. It took about 2 or 3 minutes to make all of the measurements, take the samples, and then release them. And so we did this for a period in the early part of the run in May, and then in June, July, and then in late July and August. So we had 3 separate groups that we were trying to, that we were tracking for each of the 3 years.
Is, in that machine you had there, did you go down the fish and measure fat content in different parts of the fish?
Yes, yeah, I should mention that. So there's 4 locations. We start at the head and we move down. So there's 4 separate measurements, and then we take an average to get the measure of fat. Mm-hmm. This is just a picture of the tag. It's about the size we had found. It's basically the same vendor that's been used in previous projects. It's ATS tags. And this is just, a picture of all the tools that were involved, the data sheets, all the vials to collect samples. I should also mention we take the fin clip for stock identification. This is work that has yet to be done. We're having these tissues analyzed this year, but this is the these are the vials for the gene expression work, the fat meter, and all the other tools required. These were inserted down the throat of the fish, esophageal implant. So with a PVC tool here, we were able to insert the tag in, and then the antenna extends out of the mouth of the fish. And then the tag life is about 30 days or 40 days. And we did one collection. I'll highlight some of the results from this. This was a very interesting situation where we captured and tagged fish at just below Miles Lake, just below the sonar counter in a very late part of the season in 2020. So these were basically the tail end of the run. This is the sonar daily passage count, fish and game, and so we were sampling in late July, and we were able to collect and tag about 30 fish and track them. This is the location of the tagging, and then this just shows you where Fish and Game had the radio antennas, the ground receivers that detected the fish as they swam by. So each of the major where the tributaries enter the main stem, we had receivers, and we had a number of receivers in the Volcana system as well. So those were operating through the whole season. There were Chinook being tagged at the same time, so these receivers were detecting both the Chinook and the Sockeye as they passed by. And then Fish and Game also did aerial surveys. So this is an example of a July 2021 aerial survey. And so each one of these dots is a single fish that are making their way upriver. And so, yeah, it really gives you a sense of how they're distributed over the river. And you'll see, yeah, many of them are detected close to the spawning ground.
So the plane just flies right above the river?
Yeah, and there's an antenna on the wing of the airplane. Yeah, and so they made multiple flights through the season. We had some difficulty with our tag. The tag life was not as long as the vendor told us, and so it didn't seem to affect the Chinook tags, but it affected the Sockeye tags. And so once they got up above these in the upper river, there's evidence that these tags were failing, and so the aerial surveys didn't yield really reliable results. So the results I'm going to show you are from the ground receivers that I showed you earlier. So this is the overall tagging summary. So this gives you an idea of success. We call it migratory success. So this is how many of the fish that we tagged in the lower river were successful in going through Wood Canyon, which I should mention is just above. It's in the lower river, just below Chitna, where the Chitna River enters the main stem of the copper, and it's a constricted reach. It's a very relatively difficult passage for the fish to make it through, especially under high flow conditions. So we're calling fish successful if they were detected upstream of the Chitna district, the Wood Canyon. So the key results here is that fish were not very successful in 2019. Only 20 or 32% of this is middle and late run fish were successful in making it past the Wood Canyon. In all of our other release groups, starting in 2020 and going through 2021, the rate of success was high, from 50 to as high as 80%. And so this, I think, has a lot to do with the flow conditions that I described, high flow conditions in that particular year. I should mention this Miles Lake sample was an outlier as well. We tagged 30 fish. This was that lake period. We were getting at the fish at Miles Lake. Only 2 of them made it, so a very low success rate for those fish. I don't really know why. They were not necessarily small. They weren't. They had pretty good energy content. So those results are still definitely puzzling.
Could you just throw that one out? I mean,
We may. I mean, we're analyzing each one separately. So
that's like Martians attacked that month or something, you know?
yeah, and that's a different location, different way of sampling the fish too. So we'll definitely keep that separate. I should also mention that number of tags were returned with increasing frequency as the study moved from 2019 to 2021. I think we got the word out better, and a lot of the harvesters basically dropped the tags off at Fish and Game, where I had an address on the tag as well. So some of them were sent through the mail system. So we were able to recover some of these and reuse them during the season. But as you might expect, a lot of these fish are caught in the dip net fishery. This is just an overall summary of each of the stock composition, and then comparing it with this publication from 2008 based on the early radiotelemetry project. They had a much larger sample size. They had like over 600 fish tagged in a single year, whereas we had less than 300. So this is a much better sample, and you'll see the dominant run is Clutina. That's the biggest producer of wild salmon in the watershed. This is the same stocks, the composition in 2020 and 2021. So we do see the dominant Clutina fish in the run. This is again based on where we detected these fish in the upper part of the watershed. So many of the fish were detected in the Clutina River. But yeah, you might interpret this as maybe a little bit more equal contribution of the different stocks in the river. But I caution comparison our study results with the early one, just because we had much fewer fish tagged and tracked in our study. This is what I call characteristics of survivors. So we look at fish that were either successful, yes here, or not successful, no here. So it's turquoise color are the fish that were successful, and the red are fish that were unsuccessful, that weren't detected in upstream locations. And we did see a statistical difference in size of successful versus unsuccessful fish, particularly for the middle part of the run. So the fish that were larger tended to have a higher rate of success getting up to the spawning grounds. That was true particularly in the middle part of the run, less so in the early and late part of the run. And this, of course, is this Miles Lake sample that we collected with only two fish that were successful.
So wouldn't that finding, which is obviously true, wouldn't that be counterintuitively, because if they're getting smaller due to competition in the ocean,
right?
So wouldn't evolution dictate that they would adapt to the most successful for reproduction? And that's how evolution works. But
right.
You're saying that they should be getting bigger out in the ocean if they're going to be more successful.
Right. Well, the key is if they can get that size in the ocean. So I think this is a challenge for the fish in the coming years as the conditions for migration get more difficult. And if only the larger fish tend to be successful, and they're going to suffer through increased stress and potentially what we call en route mortality, fish that basically die before they get up to the spawning ground. So that's the pattern we see in length, and we see the same kind of difference when we look at fat. So the fish that have higher fat content, especially again in this middle part of the run, and this is where most of the sockeye basically are in this run timing group, the middle run timing group. So the fish that tend to have higher fat content tend to be more successful. This is, I mentioned at the beginning, this interesting life history of Long Lake, and just by the appearance of the fish, you can see these are very different than typical spawners. This is a male and a female from Mallow Creek in the Kutina, and this is Long Lake fish. This was taken in, I believe, July. This was taken in August. So these are late run fish, and these have basically less color. They're not as red as typical spawners at that time of the year. And as I mentioned, they have much higher fat content.
And where's Mallow Creek?
Mallow is in Kutina. That's a tributary of Kutina.
So Peter, you're suggesting that the Long Lake fish never achieved that level of redness at any time?
They do. Yeah, I'll show you next.
Later on.
This is the end of September. Okay. And you'll see, they develop deeper red coloration. And many of them, I was there in late September and they're spawning, but there's records of fish living under ice basically and then spawning in the spring, as late as April. And so we're still trying to get a better handle on this. It seems like most of them are spawning in September and October, but there's certainly fish that live through the winter under ice and spawn in the spring. I was there in February. That's when this picture was taken. This is along the north shore of Long Lake, and so this is basically a spring, and it's ice-free through the whole year. So this is the water temperature. This is a logger that Sean put in. This measures the water temperature. This is through from October 2020 to the following year. And this is air temperature in Chitna. So the air temperature, as you would expect, gets very cold in the winter, but the water temperature at this site never gets to freezing. It's about 5 degrees for most of the winter during the spring. So the egg development and alevin growth and things must be much more accelerated, I think, especially for fish that are the eggs that are released into the gravel late in the season. So there must be a very high rate of development for these fish in the winter and spring period.
And do you know how long those fish are typically staying in the fresh water?
Yeah, we don't, there's been no studies. We don't know. Yeah. We don't know how long they're in and whether the timing, the smoke timing is different than other ones. And then, this isn't that clear, but we're looking at, I'm not presenting any of this information, but we're looking at fecundity, the number of eggs produced for each of these different populations, and also the size of the eggs. And it's been shown in other systems that fish that migrate longer distances have lower fecundity, fewer number of eggs, and also smaller size of eggs. And so this is Mentasta. These are eggs from Mentasta that are migrating a long distance, and they tend to be smaller than fish from Kutina that don't migrate as far. So I think we're seeing these kind of the same patterns that have been shown in other river systems.
Pete, could you go back to that Long Lake slide?
Yeah.
So there's a geologist that worked with the Park Service that said the hillside on the north side of the lake has all this water flow that's coming down and it's,
Right,
and it's upwelling up through the gravel, and that's
Right.
why that's open and why there's spawning going on.
right.
It's a very unusual situation for the
Yeah.
interior Alaska.
Yeah, I've talked to people, all over, a lot of salmon specialists, and they think, this is very unusual. Yeah, I don't know any other cases, something like this happening. So here's just overall summary of what we found. So early run fish have greatest energy reserves, and that kind of makes sense because those fish tend to travel the farthest. I didn't talk about pathogens here, but basically most of the fish use 50% of their energy reserves to get up to the spawning grounds. Both size and energy reserves can explain migratory success, particularly for the middle part of the run. And this, if you think about these results in terms of the trend of fish coming back smaller, it's definitely a lot of concern about fish not making it. And it's really hard to quantify this because, of course, you can't see the fish in the copper. There may be mortality events that happen, we just don't see them. I mentioned that other study that we published on in 2019. All of those other cases were, fish in clear water. You can clearly see fish that had died before spawning. Very hard to see that in a system like the copper. The gene expression work identifies some potentially important relationships between changing river conditions and disease, stress related to disease. So I think this is going to become a really useful tool in the future if we're able to get these samples from fish as they first enter the river. We might be able to say something about the risk of mortality of fish as they migrate up the river. So using genetic approaches like this, I think it's going to become a really useful tool.
Didn't that word, transcript, on it, does that mean gene expression?
Yes, yeah. And then this idea of, what's happening in the copper is very different from typical salmon rivers. When we get these heat wave events, it leads to a lot of glacial melting, leading to higher flows and flood conditions. And it makes passage through Wood Canal quite challenging, and 2019 was a clear case of that.
So Pete, how do you base that, or what do you base that on? I mean, is it just sort of anecdotal that you think that it's creating a barrier to passage under high flow? Or has it been actually studied?
We don't, we know where the fish, we could detect the tags from the air, when they die and the tag is motionless, it sends a different code. So we know where these fish died in the river, and many of them were observed dead below Wood Canyon in that year of 2019. So exactly what was the causal factor of the death, we don't know. We think, the water temperature was elevated a little bit, but not anywhere close to causing thermal stress in the fish. So I'm thinking conditions of passage were difficult that year, and that's a likely explanation, but it could be other things. And this linked with disease might also points toward an issue there as well.
So are you thinking that the flow is so strong that it's not preventing the fish from overcoming that strong flow, but it's just requiring more use of energy? Or is it maybe such that they just can't swim against the current at all?
I know, I don't know. It could be either of those. Really. We don't know. It would, I think, make a lot of sense to do more detailed work with, looking at their progression through the canyon. We didn't have any receivers in the canyon. But it could be a combination of a number of factors. So in terms of future work, we're hoping to, there's that earlier telemetry study I mentioned that Native Village of Diak led in 2006 and 2007. We're working with them we want to compare our results more directly with what they saw during those years and contrast migratory success and look at other things. And also look at possibility of doing this in the future, future radio telemetry work. So we're talking with Native Village of Diak to see if we can repeat this study in the future. We're working with the National Park Service, as I mentioned earlier, to try to summarize these results in the form of a model. So we now have relationships over those 3 years, and there's also a model that projects future temperature and flow in the Copper River over the next 100 years. And so what we'd like to do is try to make predictions on what migratory success is going to look like, how much energy is it going to take for fish to get up to the spawning grounds in the future, given what we're likely to see, which is higher flows and higher temperatures. I should also mention that Volcana is a really interesting system because it doesn't have a glacial influence, and so it's probably going to be less buffered with climate change. It's probably going to experience much higher temperatures than any of the other tributaries. We've already seen that. In 2019, the temperatures in the Volcana got much higher than any of the other tributaries. So I think the system is going to respond very differently in different parts of the watershed.
Is that true for Long Lake as well?
That's most of that is the Chitna, the migrating from the
But the
Chitna.
lake
So
itself does it have a glacial component in the lake
No.
itself?
it doesn't. So the lake could be affected, and the outflow stream could be affected. But the main migratory route through the main stem and the Chitna are all heavily glacially influenced. And then we're working this year now for the next 3 years on making a lot of these measurements on the same fish that Fish and Game is determining doing genetic stock identification. So they're taking fin clips and determining which genetic stock is sampled in the fishery, and we're going to relate that to some of the measures of energy and energy content and health metrics. So we're working closely with Fish and Game this year to do that, and for the next several years. And then we're trying. We don't have any support currently for this, but we're trying to do some juvenile work. Fish of juvenile fry in Cortina Lake. We're also like to do the same in Long Lake as well, and we're hoping to work with Intertribal Resource Commission to do that work. This is just just to point out the importance of getting everyone together, stakeholders, and agency people, scientists, biologists. This is the Copper River Harvesters Roundtable. Many of you may have been involved in these meetings. It hasn't happened since COVID, but it's a great opportunity. A lot of the ideas we've got for our study came from these meetings. So this is the Copper River Watershed Project that organizes this. And it's a great opportunity to, share knowledge and talk about some of these problems. And certainly, this is some of the notes that were taken on fish health. So a lot of people were thinking about, they were looking at the condition of the fish in 2019, and there was a lot of visible signs that the fish were stressed. And so there was a lot of discussion about overall fish health at that particular meeting. This was the 2019 meeting. And just want to thank the crew, particularly, Matt and Jimmy with Native Village of Diak. They run just an amazing operation with the Fish Wheel Project. They were really instrumental in making this project a success. And then Yeah, all the technicians over the years, Shane at Miles Lake has been really helpful to our project. So just want to acknowledge that. And that's all I've got. So happy to take any questions.
Do Jack Reds make it all the way up to Lake, Tenada Lake and Testa Lake?
We have not seen any jacks.
But they probably make it to Tena Lake.
Yeah, I don't, and none of the samples we've taken, we haven't seen any jacks.
Well, in one of those slides you had, I think 6 normal sized reds, and in the background looked like there were some jacks laying in the grass.
You remember, Sean, seeing any?
I'd be surprised, you know, I'd be very surprised if any jacks could make it to the upper river.
Yeah.
The lower river systems definitely have jacks.
Oh, yeah, okay.
So Pete, on one of those previous slides, the one about with the future work, I gathered that the top 3 of those bullets were funded already, and then the bottom one was not.
I lost my thing.
It sounded like you had some funding for continued work.
Yeah, so the Park Service is, the Park Service is supporting, these are, we don't have any, we don't have any funding for any future radio telemetry projects, but Park Service is supporting this work. And then this is also the Park Service. So these 2 were funded, and then this is, they have a proposal. It was unsuccessful, but we're going to resubmit that. So we're going to try to get funding for that project.
And
So
who's your hopeful funding source for that one?
that is NIFLF, National Fish and Wildlife Foundation.
Okay.
Yeah.
Have you ever visited Tena Lake?
Yes.
How do you get there? How did you get?
Yeah, Cutina Lake Road, which is a really rough.
Yeah, I tried it about a year ago today. I tried it.
Sean's been down
Had
there.
enough of that shit. So I didn't get it.
Yeah, it's really sketchy. It's really sketchy.
Yeah, that's what they told me when I got in there. I ran into.
It's fine until you get to like the airstrip, and then once you get past the airstrip,
Yeah.
it's pretty rough.
Yeah,
Yeah, I didn't, man, I think it was kind of bad that I didn't make it because I wanted to see the lake.
yeah, it's a beautiful, amazing lake.
Yeah, everybody told me.
Yeah.
Any other questions? Anybody online have any questions?
Pete, it sounds like you have some pretty good buy-in from Fish and Game on at least some of this future work. Are you working in concert with them? Are they anxiously awaiting, your results? I kind of wonder what their position is on this stuff.
Yeah, I mean, this work that we're doing with them is, on the genetic stock identification is not, it's support coming from the Park Service, and the Park Service is supporting all of these different projects. So it's not, it's basically kind of coordinated through the Park Service. So I don't have any, I mean, there's no kind of direct support coming from them, but I have, good conversations with them, and we talk about the importance of looking at stock-specific patterns. I get sort of, kind of different messages from from Sportfish. Sportfish is involved in a lot of the upper river work. And I get, so I get kind of, there's different groups within Fish and Game that I'm having conversations with. Yeah.
They let us use their chest freezers
Yeah.
Well, that's something.
and a 6-wheeler.
Yeah. and sharing a lot of their knowledge. Yeah, Mark Somerville is, yeah, really, I mean, he's been doing this work for many years, so he's been really helpful with just getting us oriented and giving us good advice. And James Alvaride in Fairbanks and Sportfish has been, yeah, he's the research coordinator at Sportfish, and he's also.
This guy behind me here, he might be helpful at some point.
Shaking his head.
I don't remember anything.
I remember you telling me about this relationship between pink salmon and sockeye growth.
Yeah.
Yeah. And that's, I mean, there's been, yeah, a lot of work that's been done on that. It's interesting.
It really is, yeah.
One of the first slides, you were talking about the North and South Bay. Is the North Bay, that's where the glacier is? There's a temperature difference between the North and South Bay?
Well, the North Bank is right above, upstream of Childs Glacier.
Okay.
So a lot of the water that's coming from the upper part of the watershed goes right along the North Bank. And then the South Bank gets Miles Lake outflow. So it's influenced by the glacier, Miles Glacier. That's how I'm interpreting it. So it's that warming signal is reflecting these warmer conditions in the upper part of the river.
And do the fish go on different sides? And what's the reason for that? Why the chicken across the road?
I know, Steve, most of the fish are on one bank, right?
Most are on the south bank.
Most are on the south bank.
And the north bank, it's really variable. Some years there seems to be a lot, and some years it's much lower.
Right.
I don't know, might just be what bank they happen to be on for the most part when they hit that narrowest part of the river.
I see, right.
Well, it seems to me like, years ago, the North Bank hardly ever had more than 10% of the count. But in the last 5 to 10 years, it seems like it's often goes way above 10%.
I think part of that is the glacier has receded. It used to be just a big cut bank, a glacier cut bank that the fish were trying to swim along on the north bank. So now that the glacier's receded more away from the river, a lot of times I think most fish are going to, you don't have a thermal barrier. That's just a hypothesis.
Yeah, it makes sense that hydrology is kind of changing as the river shifts and so on. Yeah, erosion that's back there.
Pete, as the glacial melt continues or accelerates in the upper river, what do you think the short-term and long-term impacts are gonna be on Copper River sockeye?
Yeah.
You mentioned the higher volumes could be a barrier.
Right.
Do you think that's like a long-term pattern, or how long? I don't know, yeah, I'm just curious about.
Yeah, well, the projections, the hydrologists have projected out 100 years, and so it is increase, the flows are gonna be increasing, the temperatures are gonna be increasing.
Steadily for 100 years.
Steadily for 100 years. I mean, based on that, yeah, they're accounting for, amount of glacial melting that's happening and different, climate scenarios that it looks pretty likely that those flows are gonna change. But there's also gonna be, I think, probably positive effects. I mean, I think I think some of the habitat will melt and get unlocked basically, and so fish can, be able to spawn in areas that they never did in the past. So I think there's gonna be positive effects at the same time. So that's why this kind of modeling exercise is really interesting, because I think there's gonna be, both of these positive and negative things happening.
Then you gotta ask yourself too, something like this, cold fusion is about a decade off. Which, that's one thing I do study real close.
Right.
And once that happens, it's, really oil and, carbon is really gonna just plummet to zero. So maybe the whole global warming thing will just
Right.
gradually fade. I mean, it's probably 50, 100 years, but that's gonna really change the model of speed.
Right.
But yeah, that's coming on.
Well, we're hopeful. Hopeful, but.
Hey, Pete, I've heard this theory that in Bristol Bay that some people think that the increased productivity of the last, 6 years or whatever is due to these, warming climate, that the lake productivity
Yeah.
has really increased substantially and is helping to drive these big returns. Do you think any of that is going to translate into the copper watershed?
Yeah, I guess they're finding the fry are growing faster in a warmer lake, and so they.
The environment in the ocean might be worse, but.
I know, right.
But the lake situation could get better.
Right, that's exactly right, yeah. And they think Mark Somerville talks about this, some of the lakes that are really, influenced strongly by glaciers and are cold are gonna become clearer, more productive and warmer. And so Clutina's a big producer now, but he thinks like Consina and Taslina and these other lake systems are probably gonna turn on, get more productive. And so, it's really hard to predict, but I think we'll probably see some of that going on. Unfortunately, there's no studies of the lakes, going on. I think Fish and Game used to have a limnology section, and they used to do a lot of sampling in lakes. Very little of that is being done now. That's why I'm trying to, if we can get money for the Clutina, it'd be nice to understand how that lake system might change. But that could also be an important factor.
I got a real good friend, he's a big game guy there, he works with, that's his guiding area.
Oh, right, I see some guys.
At the end with the big boats, he's got 3 or 4 boats and all that.
Right,
That's the grower. Anyway, if you ever need to get up there, I can arrange an introduction.
I think I could always use a drive.
There it is, exactly, yeah.
Is do you see an issue with Fish and Game not being funded appropriately from the state level? Does that slow down some of the research at all?
Yeah, I mean, I think that this whole idea of escapement just being measured at Miles Lake, I mean, that's really critical, but there's also this whole question of, how many fish are getting up the spawning grounds. And I know there's we are at Long Lake, so we do have estimates of escapement there, and then there's Tenada Lake, which is run by the Park Service, so those are 2 weir operations. But if you look across the rest of the watershed, there is no other comparable project. There's Native Village of Yak now has a sonar. They just started in Clutina, and so it looks like they're going to get Clutina escapement estimates now. So, and I compare it with, I did work in the Fraser in British Columbia, and they have lots of escapement monitoring going on. They have weirs set up. They have a much better handle on all of these things that I talked about, the en route mortality and the numbers of fish reaching and spawning ground. So, I mean, it becomes a question of, how much budget you have to run these. It's not cheap to run a weir. But it certainly, I think it would be a huge benefit to better understand how many of these fish are making it up there.
The department also applies aerial surveying.
Oh, right.
And they used to have a pretty good program, aerial surveys, but in the early 90s, when all the department was also funding a lot of hatcheries, and they decided to cut the budget, so the aerial surveys got reduced significantly, and all the state hatcheries got turned over to the nonprofits.
Right.
And that funding for additional surveys has never come back.
Right.
So I think right now it's like 2 rounds of surveys.
Yeah, I think they do too. Yeah. And it depends on the conditions, and it's really hard to cover all of these areas. Yeah.
Thanks a lot, Pete.